FTA and ICR. And almost he have filed 6 patent quality research publication in international and national journal of high repeats. He also was served as a secretary, Association of Food Science and Technology, India, Hisar. So, Anju, please just start your presentation, and very welcome. And as you know, we Anju is my junior also. So I was in HAU Hisar itself. I did my MSc and PhD from Hisar itself in the same department where Anju is working. So it's very nice to meet you, Anju, and please start your presentation.
Thank you, ma'am. Thank you, Dr. Sonia. And it has been my pleasure that I have given opportunity to interact on this topic. And as Dr. Sonia mentioned, we were juniors to Dr. Sonia, and we were very impressed during her time. She worked in the laboratory with full dedication. So such as seniors are rare to have, but you are inspiration to many in terms of handling the lab and the hostel issues.
Thank you.
So thank you, ma'am. And I wish a very good morning to all of you. And today we will be talking upon the role of Bacteriocin in food biopreservation. So the term is preservation. So foods, we know that it's a very perishable commodity and it needs proper storage so that we can maintain the safety and the quality of the food product. As we know that as an organism, we are human beings, we are macro one, but microorganisms, they are micro in size, can't be seen with the naked eye, but they also need the food. So whosoever win the race, that get the food. So to protect our food from these spoilage microorganisms and to maintain their freshness and nutritive quality, we need to preserve the food. And if the microbes win to get the food, then definitely it will lead to the food spoilage. And still, if we consume knowingly, unknowingly that spoiled food, then it will lead to many kind of foodborne illnesses, disease outbreaks are there. And this may be very costly and a serious public health concern. So that's the necessity that we need to preserve the food. So for the preservation, generally we process the food so that the shelf life can be extended. Simply, we can take the example of milk. If we do not process it, if we do not pasteurize it or boil it, it gets spoiled within 2-3 hours. Then to maintain the sensory properties, it is not like processing only, but we also want to retain the sensory and nutritive properties of the food. That's why we need to critically choose that according to the food commodity, which process or which preservation technology is best. Then after that, we should ensure the food safety and make it more convenient and economic value. These factors we keep in mind while we are working on the processing of the food. So there could be thermal processing or non-thermal processing. So in non-thermal processing, the major term that is chemical preservation, or we can say food antimicrobials. So food antimicrobials can be synonymically used for the preservatives, and Food Drug Administration, the US FDA, that defines the chemical preservatives as any chemical that can be added to food that tends to prevent or retard deterioration thereof. But it does not include salt, sugars, vinegar, spices, and oil extracted from spices, substances added to food by direct exposure thereof to wood smoke, or chemicals applied for their insecticidal or herbicidal properties. So we can say that they are used to prevent or retard both the chemical and biological deterioration of the food. This is a very broad category, preservative, and these should be economical. That should be used when other preservative methods are not inadequate or not available. That means if the thermal processing or other non-thermal processes that are not sufficient to preserve the food, then we go for the chemical preservatives or food antimicrobials. They extend the storage life of the food. They should not compromise with the color, odor, and flavor of the food. They should be readily soluble. And over a wide pH range, they should perform their antimicrobial properties. Safe at the level are needed. They are readily identified by chemical analysis. They should not retard the action of digestive enzymes. They should not decompose to form the compounds of greater toxicity. easily controlled and uniformly distributed in food, and wide antimicrobial spectrum. These are in general that is required from a preservative. So preservative could be antimicrobial, antioxidant, or anti-browning agent. This is the class that we are having under the preservatives. But now come the word new, biopreservation. Do we really that chemicals? Is there any method by which we can preserve our food biologically? Because the when the term biological agents or biological word comes, then a sense of security and safety come to our mind that there is no chemical, everything is biological, means organic. That is, there is will be no threat to our body. That is a common layman sense. But biopreservation, it refers to the extension of shelf life and improvement of safety of the products using cultures for protective or their metabolites. That is the biological agent itself or its products. When we use for the purpose of preserving the food, then we call it biopreservation. But why we need it? That is due to the globalization. No novel foods are there. There are many innovation technologies are there. So minimally processed food is the demand with ready to eat quality and longer shelf life. Like lot of chemicals are required or we are generally using in our day-to-day life to preserve the food, but nobody wants them. So minimally processed so that the nutrients wholesomeness should be maintained, as well as there should be like they are more safer. But there is a debate that safety aspect of chemical preservatives, so as with the biopreservatives. But chemical preservatives are responsible for many carcinogenic and teratogenic attributes, as well as they are having the residual toxicity. That's why that comes that if we have some biological agent instead of the chemical agents, so that will be preferred. So that's why the consumers tend to be suspicious of chemical additives. So the demand natural and socially more acceptable preservatives has been intensified. So this led to the possible exploitation of natural antimicrobial compounds, that is the anti against microbial microorganisms. So against the microorganisms which are occurring naturally. So then comes the biopreservation technique. We can say that it is a technique employing the natural and regulated microbiota or antimicrobials to prolong the shelf life of the food. It is capable to harmonize and rationale the necessary safety standards with traditional means of preservation and modern demand of the food safety and quality. So in biopreservation, there are lot many things. It's like first we had preservation, then processing, then chemical preservatives, then biopreservatives. So source could be any biological agent, that could be plants, that could be animals, or that could be microorganisms. In plants extract, we know that essential oils and bioactive compounds that composed of a major group that we are using in our kitchen also to preserve the food naturally. Then from animal sources, we are having lysozyme, ketocin, and lactoperoxidase. These are a few examples, but they act as a biopreservative. Microorganisms, bacteriocin that we are going to discuss today, organic acids, hydrogen peroxide, bacteriophages, and endolysins. These are the different categories, plant, animals, and microorganisms that are having the different biopreservative agents that we are going to explore. But out of this, today our focus will be on bacteriocin. So first, the microorganisms, that how they could be the source of the biopreservatives. So biopreservation is the use of natural or controlled microbiota or antimicrobials as a way of preserving food and extending the shelf life. The biopreservation of food, especially utilizing lactic acid bacteria, and we abbreviate is LAB, that are inhibitory to the food spoilage microorganism, has been practiced since early ages. At first unconsciously, but eventually with an increasingly robust scientific foundation. So with time, we come to know that these lactic acid bacteria are very useful, not only as a part of fermentation, but also as a biopreservative. Beneficial bacteria or the fermentation products produced by these bacteria are used in the biopreservation to control spoilage and render the pathogens to inactivate the food. There are various modes of action through which microorganism can interfere with the growth of others, such as organic acid production, hydrogen oxide production, resulting in a reduction of pH and the antimicrobial activity of the undissociated acid molecules. A wide variety of small inhibitory molecules that includes various gases and various organic acid. It is a benign ecological approach which is gaining the increasing attention. But they are non-toxic. That's the special attention, that lactic acid bacteria or the microorganisms are sourced as biopreservatives. They are non-immunogenic, thermoresistance characteristics, and good bactericidal activity. So lab bacteriocins are considered as good biopreservative agents. So before moving to the bacteriocins, these lactic acid bacteria, they are like, we first come to know that bacteriocins are produced, how they are produced, how they are used. Then lactic acid bacteria has a great importance in that. So they have, because of their antagonistic properties, which make them very useful as a biopreservative. When lab compete for nutrients, their metabolites often include active antimicrobials such as lactic acid, hydrogen peroxide, and peptide bacteriocins. Some lactic acid bacteria produce antimicrobial, and we know that niacin is one of the common example in that. So bacteriocins, bacteria means that are produced from the bacteria. Sins means they are having some cidal activities, killing activities. So the term comes bacteriocins, the product or the bacteria itself having the killing properties. So these are, we can define them as a proteinaceous or specific peptidic toxins produced by bacteria to inhibit the growth of similar or closely related bacterial strains. So they could be similar to yeast or paramecium killing factors, and they are structurally, functionally, and ecologically diverse. Like just like yeast having a plasmid, paramecium also produce the killing factor. Yeast and paramecium also producing the killing factors. Similarly, bacteria are having the plasmids, extra chromosomal DNA, that is responsible for various bactericidal activities. So, but the application of bacteriocins are being tested to assess their application as a narrow spectrum antibiotics. Narrow spectrums, that is not a wide range of bacteria. As in the first line, we have said that to inhibit the growth of similarly or closely related bacterial strains. And the credit to the discovery of bacteriocins goes to Andre Gracia in 1925. But he was not searching for the bacteriocin. Actually, Sir was involved in the process of searching the ways to kill the bacteria, which resulted in the development of antibiotics and the discovery of bacteriophages. So with the discovery that they discovered the extrachromosomal DNA, that is the plasmid, in the E. coli known as colicin. This is the gene colicin, and it produced the antimicrobial, antibacterial compounds to inhibit the growth of closely related Escherichia group. And the discovery is known as colicin, that is a bacteriocin produced by E. coli. These are the E. coli cells producing the colicin. So then comes that these are ribosomally synthesized proteinaceous compounds which can display inhibitory action exclusively close related bacterial species. So that's why they differ from the antibiotic in regard to the inhibitory spectrum that is exhibited by antibiotic against large group of bacteria. So bacteriocins, they are commonly produced at the log phase of the cell growth. However, there are several findings also that reveal that the secretion of the bacteriocin in stationary phase. But till date, most of them, like we can say 90% of the bacteriocin reported, they are produced in the log phase of the cell growth. So what they contain? They contain the bacteriocin, modified amino acid lanthionine as a part of the structure. That's why they are also called as lentibiotics. So bacteriocin, these are produced by the bacteria that can kill the closely related bacterial groups. But because of the presence of the amino acid lanthionine, we also know them with the name lentibiotics. So There are various efforts were made to recognize or reorganize the nomenclature of the family of ribosomally synthesized and post-translationally modified peptide. That is nomenclature of the amino, this bacteriocins, they are very diverse, but these are also the family of ribosomally synthesized, that is they are synthesized by the ribosomes, and post-translationally modified peptide, because they also include the peptides that are modified after the translation process. So these are the natural products that lead to the differentiation of lentipeptides from bacteriocin based on biosynthetic genes. That is, bacteriocin are produced by bacteria having the lanthionine, but lentipeptides are also there. They are small RIPP. Small peptides are also there. They are also having bactericidal activities. That's why these two groups are there. So these are the factors that why we promoting, what are the different factors that promote the use of bacteriocin as biopreservative? The very first thing is that they are effective in low concentration. Then they are economical, because when we have the microbial production, it is very specific and it is cost effective. Then they are safe and efficacious use of Nissin over more than 40 years in several countries. Consumer resistance to the traditional chemical preservatives and concern over the safety of existing food preservatives such as sulfides and nitrites that are specifically used in the beverage industry and the meat industry. Then advent of novel bacteriocin with broad spectrum activity of food grade, for example, lactic acid bacteria. Their activity is not lost in the presence of food additives and effective in dairy foods during storage. There are various research were made, and there is extensive application of bacteriocin in dairy industry. They are also effective under a wide range of pH and temperature, and they do not alter the acceptance quality of the food and are safe for the human consumption. And overall economical. So these are the some factors that make them very interesting for use as in biopreservatives. So how we classified these methods are 4 different methods. The very first is that how these microorganisms, that is the bacteriocin microorganisms, what is the method of killing of them, then what is the genetics, then according to the molecular weight and chemistry, and how they are produced. These are the various terms that used for the classification of bacteriocins. So like bacteriocin are characterized by what is a producing strain, common resistance mechanism, and there are large categories of bacteriocin which are only phenomenologically related. These include bacteriocin from gram-negative bacteria, colicin, and these are the bacteriocin, proteins and peptides, like what they are. If they are proteins, there are 2 categories, colicin and picosins. In peptides, we are having a large list, nisin, microsin, latrosporulin, pidiosins, plantaricin, and duramycin. Then one more classification is there, that whether bacteriocin are produced by gram-positive bacteria or gram-negative bacteria, or by the archaea. So these are the main examples of gram-positive bacteria: lentibiotics, nisin; non-lentibiotics, pidiosine PA1; large peptide, megacin A216; complex peptides, enterocin. In gram-negative bacteria, we are having the examples colicin, microcins, clavicins, pyosins, tylosins, and lectin-like bacteriocins. So these are the produced by gram-negative bacteria. Archaea group is also producing, that is sulfolobins and halocins. These are the 2 bacteriocins produced by the archaebacteria. So gram-positive and gram-negative bacteria, they are most commonly used in the in the classification system. And E. coli, in fact, that is the oldest non-colicin, is the one of the oldest non-bacteriocin, and now it is known as microcin 5. It is much smaller and produced and secreted in the different manner than the classic colicin. So we can say that gram-positive bacteria, that Nicene is known by everyone, but colicin and microcin, these are the two bacteriocin that are produced, or colicins means coli killers. That's the name is colicin, but now the name is microcin. So the naming system is a little bit problematic, but the naming bacteriocin by what they putatively kill would be more accurate if their killing spectrum were with the genus or specific species designations. The bacteriocin frequently poses spectra that exceed the bounds of their named taxa, almost never kill the majority of the taxa for which they are named. Then further, the original naming is generally derived not from the sensitive strain, the bacteriocin kills, but instead the microorganism that produces the bacteriocin. This makes the use of the naming system problematic. Basis of theory, thus alternative classification systems were also introduced. So bacteriocins that contain modified acid lanthionine as a part of their structure are antibiotics. However, efforts are made to recognize the nomenclature according to the peptides also. So from gram-negative bacteria, typically we classify them according to the size. So these are the 4 different classes. Class 1, less than 5 kilodalton. They are further grouped into type A and type B. Type A include nicene and lactocin. Type B include miracidine. Then comes to the class 2, it is more than 5 less than 5 kilodalton. It is less than 10 kilodalton, and it is having further 4 groups, subclasses. C2A, that includes pyriocin. After nicene, pyriocin is most widely used in the food biopreservation. Then saccharine A. Then second subclass is C2B, lactococin, enterocin, and plantaricin A. This comes into the subclass 2. Then class 2, third subclass is C2C. That includes lactocycline Q, gacericin A, circularin A, and canerocycline A, and C2D. That is class 1 less than 5 kilodalton, class 2 less than 10 kilodalton, having the 4 subclass C2A, B, C, and D. In class 3, that is more than 30 kilodalton, albectin and enterolysine. Class 4, that is more than 10 kilodalton. So there are 4 classes. So they are classified according to the on the basis of principal structure, molecular weight, post-translation changes, and genomic profile. Like there is not one specific criteria. So objective is on the basis of structure, molecular weight, post-translation changes, and genomic profile. So initially they were classified into 4 different classes, but later on they were then updated into 3, because class 1 nicene, class 2 plantaricin, and pyriocin. These bacteriocin are commonly described to possess significant use in food industry because of the deep sensitivity and strong inhibitory effect against the target organisms. So gram-negative bacteriocins, microcins, they are generally less than 20 kilodalton in size. Colicin-like bacteriocin, they are 20 to 90 kilodalton in size. And teleocins are so-called high molecular weight bacteriocin, which are multi-subunit bacteriocin that resembles the tails of the bacteriophages. The size and classification also coincides with the genetic structure and functional similarities. Teleocins, that is as the name indicates, tail-like. So the microcins, they are very small as compared to they are of relatively few amino acids, and for this reason they are very distinct from the larger protein cousins. The classic example is microcin 5 of Escherichia coli, and subtilocins A is another bacteriocin from Bacillus subtilis, and this peptide has a cyclized backbone and form 3 crosslinks with the sulfur. Microcins are also produced by commensal E. coli that target the other enteric pathogens such as Salmonella enteritica by mimicking the siderophores of the pathogens that use for iron scavenging. So they help in removal, or they are commensal against the enteric pathogens. Then second class, that is colicins. These are also from the gram-negative E. coli, and they are similar to the colicin-like bacteria that occur in the they target the same species and species like clapsins from Klebsiella, pesticins from Yesercia pestis, Pseudomonas genus that also produce the bacteriocin, and that is known as Piocin. S-type piocins belong to the CLB type or the F-type piocins. Colicin-like bacteriocins, they are distinct from gram-positive bacteriocin, and they are modular proteins between 20 to 90 kilodalton, and they often consist of receptor domain, translocation domain, and cytotoxic domain. And the combination of these domains between different CLBs occur frequently in nature and can be created in the laboratory. So further subclassification is there that on the mechanism like group A, group B, or their cytotoxic mechanism, whether they are nucleases, spore formation, M type and L type. Teleocins, these are tail-like. Pseudomonas aeruginosa, that is the microorganism that is very well studied, and it can be further divided into R type and F type. Some research have made to identify the piocin and show that how they are involved in cell-to-cell competition that are closely related Pseudomonas species. There are two types of teleocins, differ by their structure. They are both composed of a sheath and a hollow tube forming long helicoidal hexameric structure attached to a V-split. That's why it is a bacteriophage tail-like structure. So teleocins are coded by prophase sequences in a bacterial genome, and production will happen when kin bacteria are spotted in the environment of the producer. So they are very smart, like only when there is an enemy or a kin that they pretend them as a competitor, only then they are synthesized. That is, we can say that is depending upon the requirement. So there is a teleocin released when they recognize and bind the kin bacteria so that they can kill them. Now comes the category of the gram-positive bacteria. So the bacteriocins, they are from on the basis of their heat stability or thermolability. Heat stable bacteriocin can be further classified into class 1 and class 2. Class 1 is the modified. It includes lantibiotics and circular bacteriocin. The class 2 is the unmodified one. It includes pediocin-like, 2 peptides, leaderless, and non-pediocin-like single peptides. Then the second class under gram-positive bacteria is thermolabile. That is, they are sensitive to temperature, and it includes class 3, bacteriolysine and non-lytic ones. So these are the classification, class 1, 2, and 3. Fourth class is not there. So gram-positive, like class 1, they are the small peptide inhibitors, and they include nisin and antibiotics. Class 2 bacteriocins, they are small than 10 kilodalton, and they are heat stable proteins. This class can be subdivided into further 5 classes, and class 2A, pediocin-like bacteriocin, are the largest subgroup and contain the N-terminal that is responsible for species-specific activities, causing cell leakage by permeabilizing the target cell wall. Class 2A bacteriocins have a large potential in a food preservation as well as the medical application due to their strong anti-listeria activity and a broad range of activity. Other antibacteriocins are narrow spectrum, but class 2 are broad spectrum, and they are very effective against listeria. So one of the common example is already discussed as pediocin PA. Class 2B bacteriocin, that is 2 peptide. Bacteriocins require 2 different peptides for activity. For example, the lactococin G, which permeabilizes the increase the permeability of the cell membrane for monovalent sodium ions to get entry inside, and potassium cation, but not from the divalent cations. And almost all of these bacteriocins are found in transmembrane proteins. Then class 2C, that encompasses of cyclic peptides in which N-terminal and C regions are covalently linked. And tyrosine AS48 is the prototype of this group. Class 2D covers single peptide bacteriocin which are not post-translationally modified and do not show the pediocin-like signature. So the best example of this group is highly stable aerosine A53. This bacteriocin is stable under high acidic condition, high temperature, and it is not affected by the proteases. Then class 2E, which encompass of the bacteriocin of 3 to 4 non-pediocin-like peptides. Then class 3 bacteriocin, they are very large, but they are heat labile. So this class is subdivided into further two subclasses, class 3A and class 3B. 3A comprises of those peptides that kill bacterial cell by cell wall degradation and causing the cell lysis. The best rate under this is lysostaffin. Then class 4, these were earlier considered, but class 4 is not considered nowadays because the structural classification has, like, whether they are cyclic, like lesopeptides. Lesopeptide means they are known for their tertiary structure in intermolecular bonding, and linear structure bacteriocin like lactococin. So lesopeptides and lactococin, that is having a cyclic structure or a linear structure. Then other class, that is depending upon the so this is different classes, class 1, 2, 3, and 4. So depending upon the structure, linear and cyclic ones, they're further subgrouping. And these are the examples of the different bacteriocin that we are using in the food biopreservation, like nisin A, epidermin, PAC5, lactacin, miracidin, cinnamycin, mycin, structural incomplete. Then 2-peptide bacteriocin, pyriocin like bacteriocin, non-pyriocin like 1-peptide bacteriocin, bacteriolytic and non-lytic. And last one is the cyclic peptides. So these are the examples that are explored by the various researchers for the classification. But another classification is also there for their nomenclature based on their RIPP, that is post-translational modified enzymes. However, it is important to remark that while all the bacteriocins are RIPPs, but all RIPs are not bacteriocins. I again put a stress on this, that all bacteriocins are RIPPs, but not all the RIPPs are bacteriocins. And due to this, not all RIPPs contain bacteriocin. Therefore, only those contain that are listed here. There's first one is lengthy peptides. These are like nisin and fall under this category. Second one is linaridines, and these are characteristics with lengthy peptides, but they differ in their synthetic pathway. And the main member of this class is cypimycin. Then comes proteocins. These are the peptide of complex structure with high abundance of both non-proteogenic and deconjugated amino acids. Then linear azole containing peptides because of the presence of thiazole and methyloxazole heterocycles. So microcin is example of that. Then cyanobacteins, these are RIPs, exclusively short peptides by cyanobacteria. Thiopeptides, these are the presence of thiopeptide macrocycle alongside the abundance of dehydrated amino acid. So next come under this category is botoromycin. Botoromycins are named after the microorganisms for which they are originally obtained. Streptomyces boturopensis. This is the microorganisms. That's why the name is. Then comes the microcins and colicins. These are, we have already discussed, mainly from the Enterobacteria E. coli. And then comes lesopeptides, means the presence of a structure named lesofold. That is the interaction of the terminal macrolactam with the C ring that forms a short peptide. Then secibiotics are a subclass of the group of RIPs named after sectipeptides. They are generally produced from the Bacillus genus. And the last one is in this is bacterial head-to-tail cyclized peptides. And these are generally larger in size and their synthesis machinery, and they are representative of the class of anteriocin AS48, which shows both antimicrobial activity and other desirable biological effects. So classification is still a fluctuating topic because some prefers that how they are synthesized, some say that what is the mechanism of action. So Synthesis and mechanism of action is also plays a major role in the bacteriocin. Bacteriocin, as we know that they are ribosomally synthesized bacterial antimicrobials. So antimicrobial peptides, abbreviated as AMPs, they kill the food spoilage pathogenic bacteria, that is the target in the biopreservation. And how it acts, it forms the pores um, in the cell membrane and resulting in the desiccation of protein motive pores leading to the cell death. So killing mechanism, here we discuss about the nicene. That is the most common antibiotic we are using, that is approved by FDA, and nicene is a dual mechanism of killing in their cell. So nicene is produced by the Lactococcus lactis species, subspecies lactis, and it is commercially used in some countries. So it was first described by Rosers as a substance inhibiting the growth of Lactobacillus bulgaricus. Nicene also inhibits the growth of other gram-positive and Clostridium and Bacillus spores. And the most important quality is it is GRAS status, that is generally recognized as safe. And maximum limit of ingestion is 33,000 international unit per kg of the body weight. And there are more than 50 countries that allow their use in the products like milk, cheese, dairy products, tomatoes, canned vegetables, soup, meat, and beverage products, and mayonnaise. So it has a wide application. So other than this, that it can, after nicene, it comes the pediocin, that is produced from Pediococcus acetylacti. So how they work? So these antibiotics, first is the disruption of the cell wall, and then second is the formation of pores. This is the mechanism of action of bacteriocin. So in this mechanism, they shows the antibiotic activity, and the second mechanism is the first is the disruption of cell wall synthesis. Then second one is the mechanism of blocking the incorporation of D-glucose and D-alanine as a precursor of the cell wall molecules, and thus inhibiting the synthesis of the peptidoglycan. However, it has been demonstrated that it is also dependent upon the availability of the lipid 2 also. Once they disrupt the cell wall formation, then the second mechanism is the pore formation, that in which antibiotics conduct their bactericidal activities, and they attack on the integrity of the cell membrane. In this mechanism, two models are proposed. The first one is barrel stave model, in which the bacteriocin binds in parallel to the bacterial membrane, and this union causes, through its difference in charge, a loss of membrane potential and an accumulation of water, alongside the formation of pores that lead to the leakage of the solutes and biomolecules from the cytoplasm to the external medium. The second model is wedge model, in which the interaction of the bacteriocin occurs in a transmembrane matter, that is by the interaction of charged components of bacteriocin with the polar head of the lipid bilayer and interaction of the peptide chain with the non-polar tail of the lipid acids. And this insertion of bacteriocin generates the deformation in the membrane and fissures, and then again, it also depends upon the binding with the lipid 2. The colicin, on the other hand, this is the like disruption and then pore formation. Colicin, what they do, they actually, these are the, they are specialized in the elimination of other gram-negative bacteria, and they generally present in 3 domains that have a function. The first is antigen-like recognition section for anchoring to the cell membrane, and a mechanism like antibody. The second domain is responsible for introduction of bacteriocin to the target bacterial cell. So the formation of voltage-dependent pores in the inner membrane, the nuclease activity against the bacterial genetic material, and third is the inhibition of the peptidoglycan synthesis. So this is the mechanism of action, and there are a lot of reviews on one of the by the journal Microbial Pathogenesis, that describes that how they are classified, what is the mechanism of action, and how they behave against that. Now, how these are synthesized. So bacteriocins are synthesized as precursors. which are processed and post-translationally modified. These genes for the production of active bacteriocin are usually in the operon clusters and harbored in the genome, plasmid, or the mobile genetic elements. So it is like when there is a need, when the kin uh, uh, bacterial species is in the nearby, then this is similar to lac operon, feedback inhibition and feedback regulation is there. Then how come we isolate the bacteriocins and how they are used? So it is very important. So regarding the isolation, there are the different methodologies for the detection and determination of their spectrum, action spectrum. So after there is a good spectrum, action spectrum, then we further characterize them, and in parallel that what are these molecules and how these behave. So first is the initial assessment that a particular bacteria is producing the bacteriocin. Then there are different methods like point inoculation method, cross streak method, then radial streak method, agar insert method, diffusion method, Oxford Cup method, and diffusion well method. These are the most common basic microbiological methods that we used to isolate and screen a potential bacteriocin producing lactic acid bacteria. And these methods are based on inhibiting the growth of indicator strains caused by the presence of the strain with the possible bacteriocin production with liquid culture supernatant, and after centrifugation, these are placed in contact with the indicator strain using the various vehicles. Then aside from the conventional methods, now we have advanced methods for detecting possible bacteriocin producing strains by molecular methods, by detection of gene or gene clusters and that code to produce these biomolecules. Once the identification is over, that the bacteriocin of our interest is achieved, then extraction and purification we can proceed. Then after that, purification methods recover the bacteriocin, and finally we obtain the purified products. Then safety tests are conducted, and we also go for the different biotechnological applications. Now how these bio preservatives, bacteriocins, could be used in the food industry. There are a vast potential of bacteriocin to be used in the food component, beverage industry, meat industry, dairy industry, fish products, fruit and vegetable products. So this is a review on the application of bacteriocin in food and preservation and safety, compiling the various use of bacteriocin in different food commodities. So how this bacteriocin could be applied? The very first method, we are having the different food method, different food commodity. So that could be applied directly. They could be applied in a purified or a semi-purified form. Pre-fermented, the food product can be pre-fermented with the bacteriocin producing strain. So the very first example I am taking in the food beverage, this is the work on biopreservation of pineapple wine using the immobilized and freeze-dried microcapsules of bacteriocin producing Lactobacillus plantarum. In this work, this was done in the laboratory of CFST. In this Lactobacillus plantarum strain ATCC 8014 that was immobilized and freeze-dried to conduct the preliminary study, and they were checked against indicator microorganisms were Lactobacillus acidophilus, Acetobacter aceti, Saccharomyces cerevisiae, E. coli, Staphylococcus aureus, and Bacillus subtilis. So these were used as an indicator organisms to determine that what is the bacteriocin producing spectrum of Lactobacillus plantarum. So these are showing the zone of inhibition against free cells and the immobilized cells. Then we check the bacteriocin activity of both the free cells as such Lactobacillus plantarum, and MC is the mobilized cells. That is what is the difference in the bacteriocin activity. Then we also check the survival of these bacteriocins and immobilized cells and freeze-dried cells on gastric juices. So cymolytic gastric juices consisting of gastric juice and intestinal juices that were at different pH 4 and 7 pH, 8 pH. We have checked their potential survival. So we have found that it is effective at 3.5 pH, and there is a significant decrease in total soluble solids and sugar in wine treated with microencapsulated and fresh cells. So, but the major finding out of these research was that it does not interfere with the organoleptic, that is the taste of the wine, as compared to the free cells, and that is immobilization and freeze drying that in fact that helped in the biopreservation of the wine. This study was conducted up to 6 months, and it was found that the wine inoculated with the microencapsulated cells, the wine is more stable without any adverse effect on the organoleptic quality of the food. Then second commodity is the meat. So they can be applied as a bioconservative of the meat products. That also includes ready to eat food. So in this also meat could be direct addition in the food formula or immersion in a solution containing the peptide. Second method in the meat products is adsorption of the bacteriocin in polyethylene type plastic films or cellulose edible films or on the surfaces such as ethylene, vinyl acetate, polypropylene, polyester. Or third method that we can apply an antimicrobial coating having a bacteriocin preparation, such as lab cultures also could be applied as antimicrobial coating, immobilized in that. So to ensure the effectiveness of the activity of bacteriocin, test against the specific target bacteria must be performed on the food in which bacteriocin will be applied. But the factors like the influence of propagation of bacteriocin in the food, such as salt concentration, pH, nitrate, enzymes, solubility, lipid contact, then surface available for solubilization, uniform distribution in the food, and possible inactivation by additives. Just like in the previous, we have studied that wine, pH, and different food components that are present, how they are interacting with that and how it is affecting the sensorial quality as well as the shelf life of the wine. So bacteriocin in the meat industry, that could be used in the bovine or the poultry carcasses or in the fresh meat. Spraying the product with a combination of nicene and lactic acid. So these are the examples like different sausages, Enterobacterium fecal, target was Listeria. Cooked ham, fresh meat, dry fermented sausages, target was Listeria monocytogenes. Then this is the most common food pathogen found in the meat, smoked salmon. There are various bioprotective cultures could be applied to target the various oilage and foodborne causing microorganisms. So most common is nicene and lactic acid. So they are more efficient in reducing the count of anaerobic bacteria, coliform, and E. coli than the use of the bacteriocin alone. So furthermore, the use of bioprotective cultures, Enterobacter fecum, PCD71, Lactobacillus fermentum, and bioprotective cultures, when they have a combined application on different subclass to a bacteriocin, they are able to combat the growth of Listeria monocytogenes. However, Lactobacillus plantarum, that is a producer of the bacteriocin plantaricin, in meat that could be stored under aerobic condition at 10 degree, that is filled to control the growth of that. But when they are used in combination, they are more effective. The main limitation of the use of the nicene in the meat products is their relatively low solubility of this bacteriocin in these products, and the possibility of enzymatic destruction and inhibition of the several important pathogenic spoilage microorganisms. So that's why after nicene, pyriocin, that is produced from Pediococcus acetolacti, is the most studied bacteriocin due to the antimicrobial activity against Listeria. And its action is related to the use of this strain as a starter culture in a number of fermented foods. So it is also used in various vegetables like sauerkraut and dairy products also. So pyriocin A1 is the bacteriocin producing culture that reduce the growth of spoilage microorganisms during the storage. And the main advantage of being active at low pH, and it acts synergetically with other compounds such as lactate and organic acids that are commonly found in the meat and the fermented products. So it also reduces the count of lactobacillus sacchae in the sample of vacuum packed sliced ham. And individually or the combined effect of these bacteriocin and bacteriocin producing bioprotective cultures, respective microorganisms are isolated from the same raw material and then applied to surface of previously contaminated meat. So the beef and meat samples, they are potentially protected with the use of Nissin, and more effectively, I will say, Pediocin is having wider application. Then milk, that is lactic acid bacteria that is naturally occurring in the food, milk, dairy products, I say. So addition of the purified or semi-purified bacteriocin directly to the food, or so Listeria monocytogenes is the one of the potential threat to the spoilage of fermented dairy products, and this is the anti action of the different bacteriocin. And these are the reports that supports the study that bacteriocin could be used in the various dairy products like Nissin from Lactococcus lactis, skim milk, whole fat milk, cottage cheese, model fresh cheese. They applied purified lactine 4 to 1, reduced the Listeria monocytogenes by 3 log after 3 to 7 days at 4 degrees Celsius. In ultra high treatment whole fat milk, they combine leucosin K7. These are 80 arbitrary unit per ml, and glycine at 5 milligram per ml. They completely inhibited the growth of Listeria monocytogenes over the 7 days. Similarly, dairy food models, custard cream, UHT skim milk, then model fresh cheese, semi-purified enterocin reduce the Listeria to undetectable level in cheese within the 6 hours. So these studies are very effective, and these are the different commonly used bacteriocin used in the dairy industry. So in dairy industry, Nissin, as I already say, that is the most commonly found in the fermented, they are fermented food dairy products like cheese, pasteurized cheese spreads. In this, we can replace the nitrate for preventing the growth of Clostridia spores. And it is also effective against Staphylococcus aureus, that is the most common spoiler of the dairy products. And it also it is also effective in the refrigeration condition. And there are a variety of cheeses like cottage cheese, cheddar cheese, ricotta cheese. And Nissin is found to be effective against the Listeria. And if we use it in combination with the sodium chloride, they have produced very good results. So efficiency of combination of Nissin and bovicin against Listeria and Staphylococcus in fresh cheese was also studied. Then antimicrobial activity is also like pyriocin, lactocin, enterocin, lactococin. Along with that, genetically engineered bacteriocins are also used, but they are affected by the factors like pH, temperature, composition of the dairy product, structural, and what is the natural microbiota of the food. For example, proteolysis in the cheese making process may also affect the activity of Nissin and limits its antimicrobial activity. So protease enzyme is one of the factors that limit the use of these bacteriocin in the dairy. But if the pH values lower than 7, so Nissin generally loses activity at the higher pH. So then again a limitation that we cannot use Nissin in the dairy products. So
Hello, so ma'am, the time allotted is 1 hour. Can we take question
okay.
answer session?
Just a minute, 2 minutes,
Okay.
I will
Okay.
compile it.
Okay.
Then these are the different lab producing bacteria and products. Then in fish products, this was a study that in which bacteriocin was isolated from the fish itself, and these are the tests against the indicator microorganisms. and they have shown the incidence of enterocene structural genes, that how they are expressed and how they control the different food micropathogens. And this is how they are, bacteriocins are used in the food packaging. That is, we are not treating the food, but we can use the packaging having the bacteriocin as a component to control the spoilage microorganisms. Different fruits, meat, cereal, mushrooms, they have been treated with the bacteriocin by direct cell-free supernatant of the pediococcus. Along with the different hurdle technologies, these different bacteriocins are very effective. And then these are the leading companies. Like if you are working on that, then you can have these companies that are providing the pure bacteriocin for the research activities. These are the two databases that are available, Bagale and Bactibase, for the bacteriocin study. So Bagale is on the genomic contest. Like if you are working on the food biotechnology or molecular biology subject, then this database is very intuitive in finding the gene clusters. Then Bactibase, and this is open access designed for the characterization of bacterial antimicrobial peptides known as bacteriocin, and this is the link for the Bactibase. So these are the challenges in the biopreservatives, but in conclusion, we can say that we have discovered novel bacteriocin with unique properties, but effective application in various food is necessary in the complex environment of the food system. So this is a brief about the bacteriocins. So now the